The overall goal of this procedure is to create humanized mouse modified with chimeric antigen receptor.
This method can help answer key questions in the HIV and immunology fields in regards to testing the efficacy of stem cell-based engineered immunity against HIV infection and other chronic infections and malignancies, such as a mechanism of immune failure and ways to enhance the immune system and develop a therapy to create better antiviral or anti-tumor immunity.
The main advantage of this technique is that it is capable of a large amount of experimental manipulations. The humanized BLT mice model mimics human immunity and in this case allows for preclinical testings for cell-based gene therapies against HIV infection.
Demonstrating the procedure will be Valerie Rezek and Brianna Lamb. They are technicians from my laboratory.
Begin by using scalpels to cut the thymus into small pieces of about 1 mm cubed in a dish containing supplemented RPMI medium. Then use curved blunt forceps to place each single thymus piece into a single well on a 96 well plate. Add 100 to 200 microliters of media to all of the wells so that the tissue does not dry. Visualize the tissue under the microscope to eliminate any tissue that may not be thymus or that has connective tissue attached. Next, remove the confirmed pieces of thymus and combine them into a T25 cell culture flask.